Expertise from all over the country. At your microphone now from the Medical College of Wisconsin is Assistant Professor Dr. Robert Lochhead. He's devoted his whole career at this point going forward to this, Borrelia burgdorferi, how it leads to chronic inflammation, arthritis, and autoimmunity. Right now, he's got a focus for us this afternoon. And as they queue up his slideshow, again, a reminder that I assume yours, Doctor, will also be made available immediately.
Most of it will be, it's in review or revision, but we can talk afterwards. So first of all, this has just been absolutely fascinating meeting, and I am really glad that I get to go right after two of my colleagues, Dr. Fallon and Dr. Embers, because you've heard autoimmunity and immune regulation already a few times. And in fact, the alpha-gal syndrome, that allergy, is also an immune-mediated disease. And so I think this really sets up my story a lot. So the focus of my career really is on understanding how infection can trigger autoimmunity, and specifically using Lyme disease as a system for studying that. So I'd like to share a couple of anecdotes with you. But before we do that, I want to share a couple of slides that I give to my medical students when I introduce the idea of autoimmune diseases. So one of the core principles of immunology is the idea of immune tolerance. So what is immune tolerance? Let me see. Sorry, there's not. So what is immune tolerance? Well, our immune systems are designed to react to non-self proteins and to be tolerized against our self proteins, so we don't start killing ourselves. So the idea is, if you have a non-self antigen, say bacteria, Borrelia burgdorferi, an antigen presenting cell, which you've heard that term, so these are specialized cells that are responsible for grabbing proteins and things which we call antigens and showing them to T cells, for example. And if that T cell recognizes it as foreign, it's going to get activated and it's going to start expanding, undergoing clonal expansion. It's a hallmark characteristic of an immune response. However, if our body sees a self protein, it's programmed not to do anything, to be energetic, not to respond. So that is the idea of immune tolerance. Of course, this doesn't always work, as we well know. So this is a busy slide, but I want to show it to you because this is kind of when things go wrong, how infections can trigger autoimmunity. This is a review article from 2002, but I think it really captures nicely the complexity of the idea of infection-induced autoimmunity. It's not simple. Of course, Dr. Fallon just mentioned molecular mimicry. I don't know how many of you caught that, but that's up here on the left side. The idea of molecular mimicry is you have a foreign protein that looks like, at least part of it looks like a self protein. And so you get an immune response to that foreign protein, that T cell gets activated, but it cross-reacts with a self protein that looks like it. So that's the idea of molecular mimicry. This is the classic idea of infection-induced autoimmunity. It turns out this probably doesn't happen in most cases, certainly in some cases, but not in most cases. So there are other ideas, hypotheses out there of how autoimmunity can be developed. One is called bystander activations. Let's go on the lower left. So this idea is that there's just a local inflammatory response, making it permissive to activation, T cell activation, a lot of antigen presentation, and you just get a guilt by association autoimmunity. Another idea is up on the top right is epitope spreading. So this is a case where an infection can trigger tissue damage or cell damage, release a bunch of proteins associated with that damage, and then you get these antigen presenting cells grabbing foreign proteins, which they're activating T cell response to, and self proteins at the same time. And so now that antigen presenting cell is sending activation signals to kill the bacteria, but it's also sending activation kills to trigger autoimmunity. So this idea of epitope spreading from a foreign protein to a self protein. And kind of related to this is on the bottom right corner is release of cryptic antigens. So the idea here is kind of related to epitope spreading, but in this case, misfolded proteins or excessive tissue damage changes the structure of the proteins. And so different parts of that protein are shown to the immune system that it may have never seen before. And so you get incomplete immune tolerance to those bits of the protein. So you get like antigens rising from the crypt and all of a sudden being seen by the immune system, and now you get autoimmunity. We actually think all of these are happening in Lyme disease. So I'd like to talk a little bit more about that. Now, to simplify that slide, you really only need to focus on this. So all of those processes really can be boiled down to 1, 2, 3, 4, 5 things. All right. So first is your antigen presenting cell, like a macrophage. That expresses a molecule called MHC class 2. So I'm teaching you a little bit of immunology so you can kind of understand my talk a little bit more. So I hope you can bear with me for a minute. That is a protein that shows the immune system bits of the antigen, or the little bits of peptide to the T cell. So you have the peptide, it's a little short 9, 10, 15 amino acid piece that's being shown by the MHC to a T cell that has a T cell receptor. That T cell receptor will recognize that peptide, that antigen, as either self or non-self. And again, if it's non-self, it will undergo clonal expansion. If it's self, it should not do anything. And so what we're seeing in Lyme disease is a few different things. First of all, I'll show you some data that persistent bacterial antigens enhance antigen presentation. Another idea is that chronic inflammation leads to increased numbers of antigen-presenting cells and changes their phenotype. Elevated numbers of potentially self-reactive T cells varies widely, largely based on individual MHC molecules. So every MHC molecule presents different peptides. And then dysregulated T cell responses to pathogens impairs peripheral tolerance, which is a different type of immune tolerance. And then finally, the altered MHC2 peptide repertoire during a Lyme disease infection is dramatically altered. And that elevates inherently your risk for developing autoimmunity. So with that little introduction in immunology, now you're all experts in autoimmunity, let's look at Lyme disease. So I started my graduate work doing mouse immunology in University of Utah with Janice Weiss. I really wanted to spend the rest of my life doing this. And in order to do that, I wanted to study the human disease so I could make my research clinically relevant. So I went and did a postdoc with Dr. Alan Steer, and he and I wrote this review article a couple of years ago. So if you want to read that, the reference is there. But basically, you know, the algorithm for treating Lyme arthritis is it's more or less similar to what we've heard. So unlike, you know, neuroborreliosis or erythema migrans, Lyme arthritis is typically a late stage disease manifestation, typically 6 months or so after tick exposure. If you're not treated with antibiotics right away, over a few months, you might develop some effusions in the joint. At that point, individual says, oh, something's wrong, go to the doctor. You have this huge joint. You can see here in the center panel, those joint effusions can be extremely large and extremely debilitating. This is not a subtle phenotype, in which case you can diagnose, as was mentioned earlier, typically the antibody titers are very, very high at this point. So it's pretty good testing by antibody-based testing. Get a month of oral doxycycline, and in about 70–80% of the cases, their arthritis will resolve. But, and this is an important thing to keep in mind, I know I'm preaching to the choir here a little bit, but in about 20% of the cases, people have an incomplete response to oral doxycycline. And so a second course of antibiotics, ceftriaxone, is typically given. And that seems to be effective in resolving arthritis in another third to half of those patients that don't respond to initial course of antibiotics. So additional courses of antibiotics and changing from a bacteriostatic doxycycline to a bactericidal ceftriaxone does seem to help a subset of patients, but not all. So there's still about 10 to 20% of patients that have this persistent arthritis. And importantly, the nature of their arthritis changes at this point. So instead of these huge effusions, it becomes more like a proliferative synovitis. So this is a patient's joint inside their joint. That's synovial tissue. So usually synovial tissue is just one or a few cell layers thick. In these patients, it gets massive and it grows into the joint space. You can get tens of grams of tissue out of these patients. It's really, really quite remarkable. This looks like rheumatoid arthritis clinically. In fact, Lyme disease was initially diagnosed as juvenile rheumatoid arthritis, as I'm sure you all know, for very good reason. And as Dr. Fallon very, very nicely pointed out, at this point, you need to treat the symptoms. So as with all immune-mediated arthritides, You treat with disease-modifying anti-rheumatic drugs, immunosuppressive drugs, for example. Rare cases, synovectomy is needed to remove that inflamed tissue, but that's quite rare now that we have a really good cadre of immunosuppressive drugs. Just to show kind of clinically what that looks like, these are histopathologic staining of synovial tissue from patients. This is a post-infectious Lyme arthritis patient, so post-treatment Lyme disease syndrome. I prefer to call this post-infectious Lyme arthritis because it seems to be more of an immune-driven disease at this point. Whether there are residual bacteria that are alive somewhere or not, the jury's still out on that. But in this case, it's really driven by an immune dysregulation. And you can see a lot of purple. The purple's bad. So purple is inflammation. Those are immune cells that are going in there. And you can see the rheumatoid arthritis, which is the classic autoimmune joint disease, and post-infectious Lyme arthritis really look very similar. Osteoarthritis, which is more degenerative form of arthritis, does not look like that. So again, this kind of tells you the picture of Lyme arthritis in humans. Okay, so in that review article, I kind of proposed a mechanism of how this is happening. And basically what we thought is happening, what we think is happening is that there are retained bacterial antigens. I'm going to talk about peptidoglycan. So this is the skeleton of a bacteria, kind of keeps its shape, and it's made of like hard substance. And I'll tell you a little bit more about that in a second. But we have retained spirochetal antigens in patients. We can detect peptidoglycan in patients up to 4 years after antibiotics, after multiple rounds of immunosuppressive drugs. It sticks around for a very long time. And then we have autoantigens. So there are a number of autoantigens that have been identified, and we'll talk a little bit more about that. And basically, these two things in combination create a perfect environment for triggering of autoimmunity. This is actually an example of what we call a type 4 hypersensitivity or delayed type hypersensitivity reaction. So you heard about an allergy response. So that's type 1. For all of you who are board certified, you should know this. So that's more of an immediate response. We're going to talk about immune complexes. You heard a little bit about that. We'll talk about that a little bit later. So that's antibodies binding to antigen, forming these huge globs of antibody antigen complexes. That's another type of hypersensitivity. This one is driven by T cells and macrophages. So there are a couple of examples. So you heard mycoplasma. Like I said, this talk was really set up beautifully by our previous speakers. So mycoplasma tuberculosis caused this. So you have tuberculosis, which is this bacterial infection that infects macrophages, and our body's not able to clear it. And so we continually bombast it with an immune response, and you get these clumps of fibrotic tissue surrounded by immune cells in our lungs. And that's a type 4 hypersensitivity. Another one are rheumatoid arthritis autoantigens. So there are extracellular matrix proteins that are autoantigens in Lyme arthritis that trigger cell-mediated tissue damage. So we kind of see a lot of those same things. So really, we think mechanistically, this is what is happening in a subset of patients. All right, so I'd like to now turn to one of our big projects is the peptidoglycan story. Just want to acknowledge right away Dr. Brandon Jutras, my longtime friend and collaborator. We've been working on this together for quite a few years. And you can read this paper. Many of you may have already be familiar with his PNAS paper from a number of years ago. But what we found is that Borrelia-Burgdorferi peptidoglycan is persistent in patients with Lyme arthritis. So this is one figure from it down here showing a concentration of peptidoglycan in patient synovial fluid. I'll say of the 34 patients, I think like 29, this was after at least one course of antibiotics. So most of them had already taken antibiotics before we ever saw them in our clinic. Synovial fluid from control patients, rheumatoid arthritis and stuff did not have it. Lo and behold, if you inject it into mice, they get arthritis. So it's an arthritogenic compound that persists in patients for years. So that's really interesting. So another observation that I made as a postdoc is that in our synovial tissue, so this is an example of synovial tissue from patients. And what I've done is I've stained for MHC class 2 molecules. So human, the most abundant is called HLA-DR. You don't need to memorize that, but these are the molecules that show that antigen to T cells. And you can see that green is really, really bright in there. So it's a lot of antigen presentation, typically of a type 4 hypersensitivity reaction. Red stands for stromal cells. So these are fibroblasts. These are not immune cells. These are structural cells. The yellow are those fibroblasts that are expressing MHC class 2. So for the immunologists in the field, you should know that fibroblasts are not antigen presenting cells. Macrophages, dendritic cells, B cells are. So that was really interesting. We actually validated that outside and in mice. So fibroblasts can indeed, under certain inflammatory conditions, express MHC class 2. So the burning question that we had as we started out my lab at Medical College of Wisconsin was to ask whether these fibroblasts are acting as tissue localized antigen presenting cells, driving this delay type cell mediated hypersensitivity reaction. So my wonderful graduate student, Joe Rouse, who's going to shortly be joining the CDC working on influenza vaccine work, did a number of great experiments. This is just one experiment that I pulled from that. And basically what we did is we took fibroblasts from mice. We stimulated them with conditions that we see in these Lyme disease patients. We give them interferon gamma, which triggers this MHC2 expression. And then we give them peptidoglycan, like we do see in post-infectious Lyme arthritis patients, or we don't. And what he found is that, and then he put special T cells. So these T cells recognize one antigen and one antigen only, and it's this little bit of protein from chicken albumin. So it's used a lot by immunologists to test antigen-dependent T cell responses. All right, so with this, and it's highly artificial system, but what we wanted to test is whether these fibroblasts were able to elicit this activation, this clonal expansion of T cells, like I showed before. And lo and behold, on the bottom left, sorry, you can't really see that because of the line, but you can see if we pre-treat our fibroblasts with interferon gamma, you get some T cell proliferation. If you add in peptidoglycan with interferon gamma, you supercharge these fibroblasts. So now they're going great gangbusters. They're really good at activating T cells. If you put in a different peptide from chicken albumin that's not recognized by these T cells, you don't get any response. So this is specific to that bit of protein presented by that MHC class 2 molecule. What's more, on the bottom right, you can see that not only is there a difference in the number of T cells that are proliferating, but the rounds of T cell proliferation are vastly different. So in the fibroblasts that are just given interferon gamma, you basically get T cells proliferating once, and that's shown in the bottom right-hand corner. I don't know if you can see that, with just one hump. So I wish I had a laser, which I don't. But right here, these are cells that are not proliferating. So we give them a dye before we do the experiment, and they're bright. And then every time they divide, they lose that brightness by a half, all right? So this peak right here, cells have divided one time, so they have half as much of the dye in them. And then that, you can see that kind of peters out. However, oh, thank you. The lower part, which none of you can see except those on the front row, you can see that looks very different. So you've got multiple rounds of proliferation. T cells are proliferating, and that's kind of showed graphically here. That indicates a very robust T cell response. Clonal expansion, we see a lot of inflammatory cytokines produced by these. So this really demonstrates that yes, these cells are antigen-presenting cells under conditions seen in Lyme arthritis. So the kicker is that you need that peptidoglycan for this to be really robust, which we see in our patients. So this is kind of the model. And like I said, this is analogous to tuberculous granuloma in tuberculosis, which is triggered by persistent antigens of Mycoplasma tuberculosis. So that's kind of a really big story. We're getting that submitted. We're getting that published shortly before he graduates. Oops. However, this isn't unique to Lyme. So we actually noticed that in some of our control patients, we could detect peptidoglycan not Borrelia peptidoglycan, but other types of peptidoglycan. And so we actually, I did a study with a colleague who's an orthopedic surgeon, collected synovial tissue samples from these patients who are undergoing knee replacement surgery, no Lyme disease. This is just, run-of-the-mill knee replacement surgery. And look at this. You see the brown dots? That's bacterial peptidoglycan in their joint tissue. And at the bottom, you see that H&E staining, and that purple is bad, remember? So that's inflammatory cells. The pink rings, that's fibrotic tissue surrounding those pockets of peptidoglycan. So this clinically looks like a tuberculous granuloma, type 4 hypersensitivity reaction. This is what we think is happening in Lyme disease and possibly in other types of arthritis. We're still pursuing this a little bit more. So the take-home message of part 1 of my talk is that uncleared or persistent bacterial peptidoglycan may trigger granulomatous type hypersensitivity reactions in synovial tissue. That's a lot of really tricky immunology words, but I hope now that you're experts in immunology, that you can kind of understand now mechanistically what's happening in the joint. All right, human observation number 3. So moving on to the second part of our talk. So as I mentioned, 7 Lyme autoantigens have been identified in humans, and this is not rare. So half of patients have autoimmunity to one or more of these autoantigens. So in Lyme arthritis, it's very common. They group into 2 categories. So we have 3 that are proteins involved in vascular damage or vascular inflammation. Apolipoprotein B100, which we're going to talk about more, endothelial cell growth factor, which Dr. Fallon mentioned briefly, and annexin A2. 4 of them are extracellular matrix proteins. So these are proteins that make up the tissue of the joint. Fibronectin, which I'm going to talk a little bit about, collagen, laminin, and a matrix metalloprotease. So the other thing I want you to take home from this slide is, so Dr. Fallon, again, this is brilliant. I don't need to talk about COVID, but so he talked about these microvascular lesions in long COVID, right? We see the exact same thing in these patients. It's a damaged microvasculature. You see these little lesions, and you can see on the left, on the top, H&E staining. I've zoomed into blood vessels. On the top, you can see those purple cells surrounding that little blood vessel. That blood vessel is about ready to explode, get destroyed by those immune cells. And even intact vessels on the bottom, you still see quite a bit of inflammation around those blood vessels. In fact, you can see with again, looking at antigen presenting cell, those blood vessels are surrounded by antigen presenting cells. So again, this indicates that the microvasculature is a major target of your immune response. And in fact, for ECGF, you can see one of those autoantigens accumulating in those obliterative microvascular lesions, really demonstrating that the autoimmunity to these vascular proteins are triggering vascular damage, like long COVID. What's going on here? All right. Human observation number 4. No, wow. Okay. Borrelia-Burgdorferi outer membrane proteins and molecules interact with these autoantigens, either directly or indirectly. So there are a lot, this is a table I put together. There are a lot of proteins, outer membrane lipoproteins, people much smarter than I have identified a lot of the binding partners of these Borrelia proteins. They include glycosaminoglycans, laminins, collagens, fibronectin. The Borrelia proteins bind to these host proteins, cause damage to these proteins that then become autoantigens. There are a number of vascular associated proteins that are targets of Borrelia proteins. So I don't think this is coincidence. I think the interaction between Borrelia and these proteins are somehow triggering autoimmunity. So that's kind of the other part of our research, is trying to figure out mechanistically what's happening. So we've moved to mouse models to try to figure this out. And so Dr. Embers graciously talked about IL-10. So that's an anti-inflammatory cytokine. Mice lacking IL-10 develop this massive immune response that's driven by T cells. So this is a great model of kind of T cell-mediated Lyme arthritis. Autoimmune-like, all right? And then your normal run-of-the-mill black 6 mice, they develop mild, transiently inflammatory Lyme arthritis, ho-hum, which is great. So we have 2 comparison groups. We have one that develops this type 4 hypersensitivity sort of thing, one that doesn't. And all we did is we said, all right, what are those peptides that are being presented by MHC class 2 molecules during infection? And are they changing over time? And if we're really lucky, can we look at some of those autoantigens and see if there are changes? So that's what we did. COVID and years later. So this would just very briefly, the size of the peptides are about what one would expect to be bound to MHC class 2 molecules. We did this. We found thousands of peptides from every mouse group. We did uninfected mice, and then we did mice at 4 weeks post-infection, which is the peak of arthritis in like the black 6 mice. And then at 16 weeks post-infection, where arthritis is fully resolved in the black 6 mice, but continues in the IL-10 knockout mice. And the stuff on the right says that the nature of those peptides are consistent with what one would expect. So this is kind of validation of our data set. And so what are some of the proteins that we source proteins? Well, this is kind of a set of protein classification of the peptides that we found. And I just like to point out that cholesterol metabolism comes up as the top or second top in several of those groups. A lot of proteins involved in cholesterol metabolism ended up being increased in the draining lymph nodes of these infected mice. We thought that was interesting because one of those autoantigens is ApoB100. What is ApoB100, you ask? Well, how many of you have heard of LDL cholesterol? Okay, ApoB100 is the dominant lipoprotein in LDL particles. It is an autoantigen in about 50% of patients during infection, Lyme arthritis patients. It's also an autoimmune target in atherosclerosis, both in mice and humans. So keep that in mind. So in uninfected mice, both black 6 and IL-10, we found one peptide basically from this huge, it's over 4,000 amino acids large, one peptide. So it's really not presented very well. It's not efficiently presented. All of a sudden, when we infect mice, you get this huge expansion of the number of peptides from ApoB100. So this is an example of exposure of cryptic antigens. And we were lucky, one of these peptides, P2 here, so P1 in mice is not immunogenic. It's known to bind well to MHC, and you don't get a T cell response to it. Peptide 2, however, is a known autoantigen in murine atherosclerosis. So mice in a certain genetic background, given a high-fat diet, they develop autoreactive T cells against that peptide, P2 peptide. And we only see that in infected mice. So we think that what we're seeing is the beginning stages of cryptic antigen exposure during an infection of this lipoprotein. Well, we thought that was interesting. And then this paper came out in May. Cholesterol contributes to risk, severity, and machine learning-driven diagnosis of Lyme disease in May. So I encourage you to look this paper up. I thought it was really fascinating. And I'm just going to literally read a little bit of their results. Increasing cholesterol was associated with a greater risk of Lyme disease, and hypercholesterolemia was more prevalent in Lyme disease cases than in controls. Cholesterol genetic scores and rare loss of function variants in CD36 and LDL receptor were associated with Lyme disease risk. Serologic profiling of cases revealed parallel trajectories of rising cholesterol and immunoglobulin levels over the disease course, including marked increases in individuals with loss of function variants, so familial hypercholesterolemia and high cholesterol genetic scores. So wow, humans with high cholesterol are more at risk of getting Lyme disease and have an increased risk of developing more severe disease. Well, that's interesting. Guess what one of the most dominant components of Borrelia inner and outer membranes are? Cholesterol. They can't make it, they have to steal it from their hosts. So they're stealing cholesterol. I don't think that's coincidence. They need it to replicate, and these cholesterol binding lipoproteins are common autoantigens. So this is some brand new data, unpublished. I'm going to have to delete this from our slides because this is preliminary. We're still figuring out what's going on, but I just had to show this to this audience. So we took the classic hypercholesterolemic mouse model. It's an ApoE knockout mouse, fed them on a high-fat diet, cheeseburger diet, gave them Lyme disease, waited 15 weeks. And this is what we saw. So these are on this, in the center are under uninfected controls that are fed that high-fat diet. And I talked about synovial thickening. Right here, this little bit right here, that's what healthy synovium looks like in mice and humans, just a little few cell layers thick. That's a tendon, then you have the synovium surrounding it, and then bone looks like that on the top, healthy bone. I want to show that to you because in our infected mice, so on the left, and this was penetrant, all 5 of our infected mice had this, none of our uninfected mice had it. We see massive tenosynovitis targeting the tendons. We think that that's important. So you can see now that that synovial lining is very thick and very purple. Purple bad, right? We see pannus formation. So you don't know what a pannus is, but it's very common in rheumatoid arthritis. Bony erosion. So this was seen in 2 or 3 of the mice. We actually see destruction of bone. That is irreversible joint damage. That cannot be fixed, no matter how many courses of antibiotics you give these mice. That will never be fixed. Those purple dots around there are all plasma cells. So Monica Embers talked about plasmablasts. Plasma cells are B cells that are making antibodies. You have synovial villi growing into the joint space. You saw the picture of that patient with those kind of villi things. It's exactly what we see in Lyme. In fact, this is very reminiscent of mouse models of rheumatoid arthritis. We also do that in our lab. And those mouse models are driven by immune complexes. Dr. Fallon also talked about immune complexes. So that's type 3 hypersensitivity reactions for those of you paying attention. So immune complexes, these big antigen antibody complexes against soluble antigens. So these are oftentimes antibodies can become autoantigens in rheumatoid arthritis. Damaged neuronal proteins, evidently in Lyme neuroborreliosis, DNA in the case of lupus. These are examples of antigens that form immune complexes. So these big globs get stuck in the blood vessels, in the extracellular matrix, and cause chronic inflammation. And this is exactly what happens in mice when you give it to them. So it's autoimmune-like. Now, is it autoimmune? I don't know yet. We're doing those experiments, but it sure looks like it's immune mediated and particularly B cell antibody mediated joint inflammation. What's more, if you can see on the bottom right, these inflamed tissues contain what look like cholesterol crystals that accumulate in the joint. Now, a colleague of mine, when he was doing the ApoB autoimmunity paper, he noticed really, really high levels of lipoproteins in patients, Lyme arthritis patients, both during and after infection. I don't know what that means, but I think it's really interesting. Particularly, and the last thing that's hard to see in this picture, but we actually noticed something really funny. I have never seen this in mice. I have done thousands of mouse infections, given a lot of different mouse backgrounds, Lyme disease. I've never seen this before. These are bumps on the surface of near the joints. So this is seen in rheumatoid arthritis. They're called rheumatoid nodules. But when we look histologically, we see bumps along the tibia of our mice. So it's weird. We get these what look like rheumatoid nodules on their joints. So again, hinting that there's something really immune mediated, possibly autoimmune going on here. So of course, these are hypercholesterolemic mice. So they also get atherosclerosis. Uninfected mice do as well. We see these sheets of plasma cells within the arterial plaques, lipid cores within them. So just like I saw in the joints, look like atherosclerotic plaques. But again, both uninfected and infected mice get this. So I don't know. I'm not a trained cardiologist, so I can't really analyze these carefully yet. I'm getting those done, which is why it's still preliminary, but I see kind of clinically, phenotypically, these lesions are quite similar. But if you look at other parts of the heart, you do start to see some marked differences. So for example, at the base of the heart, you see inflammatory cells within the stromal tail, so this fibrotic tissue. You start to see inflammatory cells in the infected but not the uninfected mice. You also see marked inflammation of the aortic valve. So that is not a subtle phenotype there. So these are the valves, the valves that allow blood to go from the ventricle up to the aorta, and we really saw massive inflammation. These mice are really sick. In fact, we noticed it. They were lethargic. They lost weight despite being on a cheeseburger diet. They couldn't walk well. There is something really wrong systemically with these mice. So we're chasing that. We also see mixed leukocytic myocarditis. That's a big word, but basically what happens is you get these like basically scars in your heart muscle with immune cells, and then your cardiomyocytes get enlarged and inflamed, and that's what we see. I'm not a Lyme carditis expert. I have a couple papers where we looked at this. A lot of this is similar to what we see in other models, but mice do not have Lyme carditis at 16 weeks post-infection. They get it about 2 or 3 weeks and then it goes away. So this is highly unusual. This is a case of chronic, what I think is chronic Lyme carditis. I don't know yet. We're doing some time courses, but I just wanted to show this to you as like some new exciting data that is consistent with our other studies and with that human observation of cholesterol. So just the last little thing, I'm bringing it back now. So in our immunopeptidomic study, we also found fibronectin. We found some that are actually identical to fibronectin proteins that are found in human patients. That's highlighted here. One of them is a known autoreactive T-cell antigen in Lyme arthritis. So that's kind of bolded there. That's the peptide that was pulled out of a patient that was tested, and you get autoreactive T cells against it. That's shown on the left. The mouse fibronectin peptide we found was right here. And there's a protein from Borrelia that looks a lot like it. I have it in a box, this pan-K. It's not highly conserved, so it's not the same in all Borrelia species. But we noticed that if you get a T cell response to this peptide from fibronectin, you also get a T cell response to that Borrelia peptide. Is that a case of molecular mimicry? We don't know. It's possible that we could be seeing an example of molecular mimicry. More research is needed, though. And that exact same Lyme autoantigen from fibronectin is presented by Lyme fibroblasts from human patients. So coming back full circle, the peptidoglycan and the fibroblast as antigen presenting cells, if you stimulate fibroblasts from, this is in human patient now with Borrelia burgdorferi, they express this peptide that's a known human Lyme autoantigen from fibronectin. So there it is. So they can present Lyme autoantigens in vitro, just like we've identified in patients. So a nice side effect of this immunopeptidomic study is we've actually identified quite a few Borrelia proteins that are presented by MHC class II molecules. So this is the list, and you can kind of see actually we're getting some peptides that we're seeing over and over again in humans and in mice, interestingly enough, in different tissues, different cell types that we're studying. So we think we're getting to a point where we're kind of seeing what antigen presenting cells are what Borrelia proteins are showing antigen presenting cells. Now, I don't expect you to know all the Borrelia proteome, but if you scan through there, you will notice that none of them are outer membrane lipoproteins. Those are the immunodominant types of proteins for B cell responses. We don't see them being presented on MHC class II. I don't know what's going on, but it may be that this is one of the ways Borrelia are suppressing our B cell response, because B cells need this antigen presentation to do a really robust job. If they're not seeing these peptides on MHC class II, then they're not going to have a really good high affinity antibody response. That's pure speculation on my part, but it's really interesting, and it can't be coincidence. All right, I'm going to skip this for the sake of time, but this is kind of what we found, is that Borrelia interactions with these proteins tend to trigger autoimmunity. Damaged extracellular matrix causes a delayed type hypersensitivity response, particularly when you mix in peptidoglycan into the joint. And that probably triggers autoimmunity, but also peptidoglycan acts in bystander activation sort of way to enhance, make it a more permissive environment for autoimmunity. And the vascular autoantigens seem to, we think, trigger immune complex formation. And why I think that's interesting and important is because unlike the delayed type hypersensitivity, which is really localized to an affected tissue, immune complexes are systemic. So it's going to hit the vasculature. It's potentially hit the meninges in the brain, vascularized tissue in the heart, in the joints. So I really think that this idea of immune complexes may be really important, not just in Lyme arthritis, but potentially in autoimmunity and other Lyme disease manifestations, particularly patients with post-treatment Lyme disease syndromes. That's me speculating, but that's kind of where I think this is going. And my colleagues, my collaborators, students, particularly the Jutras lab, we've worked together a long time and continue to do so. And my funders, thank you very much.
Thank you, Dr. Lochhead. First question, front row, Brian.
Thanks, Rob. Interesting talk. I want to kind of go back to your pan-K peptide or your sequence there that you said was not conserved per se across Borrelia species or strains. This is JD1, so it's a well-known characterized burgdorferi species. Is this differ among the strains of Borrelia burgdorferi?
Yeah, N40 or B31. There's some differences there.
So how about like an Epitheliae?
I did this like in like 8 o'clock at night last week and found this hit and threw it in here. I don't know, but that's it's actually the same one that we studied in that JCI paper, Kanjana study. But you know, they're actually, if you go read this paper, this is not the only potential molecular mimic of extracellular matrix proteins. I think there are 4 or 5 candidates that we were kind of looking at. This actually is one of the ones with weaker data suggesting molecular mimicry. There are some that are stronger associations, but that's the one we found in our fibroblasts and in our mouse. So, yeah.
And if I could do one more, the peptidoglycan. Borrelia notoriously has low cell density in an infection, and that means low peptidoglycan levels. Would you expect a dose response? Think of another infection that may has higher levels of bacterial cells and therefore higher levels of peptidoglycan left over.
Yeah, so you know, Borrelia is a gram-negative bacteria, so it has thin peptidoglycan. Staph aureus is a gram-positive, so it has a thick layer of peptidoglycan. So it's the classic peptidoglycan, and you know, studied. You're right, but something that's really interesting is that Borrelia cannot recycle their peptidoglycan. So as they replicate, they shed it. And they can't reuse it like most bacteria. So as the cells replicate, they're shedding it. So if you're infected for, 3, 4, 5, 6 months, that's accumulating, right, over time. And we think that's important. The other thing, it's not published yet, but it seems to be refractory to degradation because of its unusual structure. So we're looking at that actively, but we actually think it may build up to quite high densities. We don't know that yet, but stay tuned. Brandon's really focusing on that, so I can't speak too much on it, but it's weird. It's different, and it does it differently. Shocker. Okay.
Hi, Lauren Edline, SUNY Upstate. In patients with pre-existing mast cell activation syndrome, I'm thinking of a couple of my patients with Ehlers-Danlos syndrome here. Do you think that would have an impact on whether or not they develop autoimmunity after infection?
Yeah, that's a really good question. I was thinking about that as you're going through. So that's typically more of an IgE mediated response. We really don't see much of that, at least in Lyme arthritis. It's more IgG4 and IgG2. So more of your isotype switched IgGs. So I don't know is the short answer, but it looks more like immune complex or cell mediated, kind of not mast cells. But that's arthritis. It's going to be different for different manifestations. Monica.
Okay, so I'm really interested in this hyperglobulin or hyperlipidemia. There's a researcher named Bruce Patterson who's studying long COVID patients and treating them, and he basically studies immune biomarkers, and he's treating patients with statins. And he also claims that this is going to work well with Lyme patients. And so what I've learned is that statins don't just reduce cholesterol, but they also stimulate a pathway that reduces vasculitis. Is your next step basically to take in mice and treat them with statins?
Which study section are you on? Absolutely. And not just statins. There are a number of ways you can lower cholesterol. So we're looking at a few different ways. Yeah, absolutely. I mean, it's a no-brainer, right? Absolutely.
You characterized a lot of antigen proteins. And what are the implications of that for developing a vaccine against Lyme disease?
Yeah, that's a wonderful question. So basically what we want to, an inherent problem, not just with Lyme vaccine, but COVID vaccine, a lot of vaccines, is you don't tend to get a good antibody response, but sometimes you don't get a great T cell response. And so the duration of the vaccine efficacy is lower because you need that T cell help to kind of kick it in gear, and you have to make it strong. Neutralizing response requires T cells. So our thought right now, our plan is to conjugate some of these good T cell epitopes with B cell antigens that are known, OSP-C, OSP-A, et cetera, et cetera. And what that would do in theory is hyperactivate the B cell response to those outer surface lipoproteins, generating higher, longer memory, broader protection, things like that. So in theory, you would require fewer boosters and it would last longer and be more cross protective. So it's being looked at in COVID, for example, vaccine. And so this is kind of the way we're thinking about this, is not a standalone vaccine, but more to be used in conjunction with some of the vaccines that are in clinical trials right now as an add-on. Great question.
Are there more good questions? If not, how about a round of applause for a doctor from Wisconsin, Dr. Rob Lochhead.